Hello again. So now we're going to carry on with carboxylic acid salts. So we already said carboxylic acids. So anything with this kind of a group on one of the ends, so a double bonded oxygen with another oxygen on it, is pretty willing to get rid of that hydrogen. And so what we would end up with is a negative charge on that group, and then probably some kind of counter ion. So here we just have, if we're reacting it, and it doesn't need to be with a strong acid or a strong base by any means, it can just be with water to a certain extent. Like any kind of weak acid, it's going to have some deprotonation in water. But with a strong acid like sodium hydroxide, it's definitely going to be fully deprotonated. And what we end up with is this negative charge that's now shared across these two oxygens here. We'll have a slightly positive charge over here. But really, it's going to be balanced with some other positively charged ion, like the sodium ion in solution. So what does that mean? So if we have a carboxylic acid, it can have a very long carbon chain. And this is nonpolar in its entirety, right? You can see it looks more like oil and gas. It's not going to mix very well with water. But the head group over here, this is polar and is quite soluble in water. So where we have hydrophobic, tail, okay, so hydrophobic meaning just it's not soluble, it doesn't like water. And of course, on the other side, we have a hydrophilic head group, okay. The molecule overall is what we would call amphiphilic. Holy cow, that's terrible to write. Amphiphilic, meaning kind of parts of it are going to be water soluble and parts of it are going to be soluble with grease or with non-polar kind of objects. So this would be sodium stearate. So the 8 just means that it's a negatively charged group like that, or the conjugate base to an acid. We've talked about that earlier in the course. Sodium, of course, just because there's sodium attached to it. And this would be otherwise called, I guess, stearoic acid. We could count up the carbons and come up with the standardized name if you want to, but usually it would just be called stearic acid. So S-T-E-A-R-I-C, stearic acid, as one of the more traditional names for it. And the function for a molecule like this is a soap. So just as a general soap, what you do is take a fat or something with a long carbon chain like that, break it into carboxylic acids or oxidize it to get the carboxylic acid, react it with a strong base, and you end up with this polar head, non-polar tail group, and it's a detergent. The non-polar part will bond to, or at least encase, non-polar objects like grease, something that water wouldn't dissolve, and the watery part dissolves it in water. So what we end up creating is what's called a micelle of these. So if we draw them as just kind of like the head group, the hydrophilic tail, what they'll do is they'll encase like a pitch of, like, I don't know, a ball of grease. The non-polar side will be attached to it. The polar part is now water soluble. So this whole thing now becomes water soluble and it can wash the grease away. So that's great if we're talking about like washing dishes. Right now, the focus for why we care about soap is something slightly more critical. So say coronavirus, for example, as something we might want to destroy with soap. Why is this going to work? So here we have a very basic model I just grabbed off the internet of an enveloped RNA virus. Okay, so we've got these proteins on the surface. This is what's going to bond to our cells to kind of open the door and let it be taken in by the cell. So you can see, yeah, so we've got attachment to the target cell required for virus release, and something else just to create a round envelope of the for the RNA. So this is the part that's actually causing all of the damage, that's self-creating, okay? But this host envelope, so these phospholipids, so this is the same idea. So it would have phosphorus in the hydrophilic head and lipids, so just hydrophobic tails, so quite a lot like the soaps that we were just talking about. This is actually the same envelope as our cells, so this is us that it's taking and incorporating into its own envelope. But because it relies on this same hydrophilic hydrophobic molecule to create this envelope, what soap does at the right concentration and for the right amount of time, like follow those washing directions, is the soap molecules will be able to get into that envelope and destroy the envelope. So break up what are these bonds here, and really take each of these proteins and now to isolate them and destroy the envelope and destroy the virus. So that RNA can't get into the cells, it can't release itself, it can't self-replicate. It just absolutely destroys this molecule. We don't need to think about, say the, let's just move that directly inside. Okay, that's the soap working. We don't need to think about a virus as some sort of complex living thing. We can think about it as just a nanoparticle, just a very cleverly designed nanoparticle, and one that is more chemistry than biology. So just with some very basic, very old school chemistry like soap, we can destroy something that is a very, very advanced, if we were trying to put this together in a lab, nanoparticle. So that's going to be it for right now. Just enough to say, basically, wash your dang hands, keep yourself safe, and to trust soap to get the job done. Next video, we're going to move on with esters and amines and amides, and that'll cover it off for functional groups. Okay, so everyone, we'll see you next time.